Hi, this is my operon video, so I will get started. So here we have the trip operon, and this is the promoter, which is where the RNA polymerase is going to recognize a sequence and bind to start transcription. And then this is the operator, which is where the repressor protein can bind. Then all of these are genes, which are going to help in the production of enzymes, which are going to make tryptophan. So it all starts when RNA polymerase, it's going to bind to the promoter because it recognizes a DNA sequence there, and it'll start transcription. It'll go over the operator because there's no repressor there. Then it'll go over all of the genes, which is going to cause those enzymes to produce, which will synthesize tryptophan. So then, that's going to happen a bunch of times. And then once there's enough tryptophan in the cell and it doesn't need any more, it's going to bind to a repressor protein, and that's going to cause the repressor protein to bind to the operator. And then when RNA polymerase recognizes the sequence, when it recognizes the sequence of the promoter and binds there, it's going to try and keep going down, but the repressor protein is going to block it. So that's going to stop the transcription, and then no more tryptophan will be made. So for the conclusion questions, how are inducible and repressible operons different? So this trip operon is a repressible one, which basically means that it's usually on, but it can be stopped from functioning when the repressor comes. It'll be repressed. But then inducible ones are ones that are usually off, but it can, a small molecule can come and bind to it, and it'll cause it to turn on and synthesize. And then how is it beneficial to be able to turn them on or off? Well, so for this one, when there's enough tryptophan in the cell, it's going to be unnecessary to waste energy and materials to try and make more. So it's basically conserving energy, and it's making sure that the cell only has what it needs at that time. Then what would happen to the amount of tryptophan being produced when the repressor is attached to the operator? So whenever this attaches, it's going to block it from producing any of these enzymes that are needed to synthesize tryptophan. So the level in the cell is going to decrease, and basically, because that'll only happen, it's only going to bind whenever the levels are high in the cell. So that means that then over time it's going to decrease. So no more is going to be produced, and then eventually it'll get low enough that it's going to just synthesize more. What would happen to the amount being produced if RNA polymerase were destroyed? So this is the RNA polymerase, and if it got destroyed, it basically wouldn't be able to bind to this promoter sequence, and that's what starts the whole process. And it would essentially be the same as if this repressor protein malfunctioned and it couldn't come off. No more would be produced in the cell, so tryptophan levels would get really low, and it probably wouldn't be able to do all its metabolic processes that require tryptophan. So I think that was all of the questions, but basically it's going to start, recognize the promoter sequence. If the repressor's there, it can't go. If it's not there, it's going to go past the operator, go down all of the genes, make those enzymes, then tryptophan will come, and that's basically it.